How is everyone this morning?
Doing best, someone.
It's very challenging to come halfway around the world and then try and focus and teach and concentrate, because right now, Pantelis and I, what time is it?
Ready to go to sleep.
Yeah, it's just, so normally our bodies are telling us it's time for us to go to sleep. So in the United States, our families are just going to sleep.
진짜 다 zambuling chega dus kora kon le rawas deza. Lebat lega shira che chimindus. Namgyu imbe na ta tanda ma niga dwe ju res ngonzo. Tanda ngonzo subu zo. Ani tanda nieg thadi che dig rawas. Tanda del sam rigwa dimchi lega che de khapudus.
So it's interesting biology behind the so-called circadian rhythm. So your body gets into kind of a 23, 24-hour pattern, and all the genes and all the cells are moving in this kind of cycle. So your body gets set that way, and then when you leave that rhythm, it becomes much more difficult, and your body has to reset that clock. So it's all going on in your cells and your brain. It's a very interesting biology we'll talk about one day.
제가 디게 아니 겸종게 단다 만주다 류기 수국 레가치다 냐망아주 페제 깨제 신유여 니샤 투지기 아니 콘드스에치 고라고르단치 만주고 수국 나로 예베게 아니 직다 차시 간데 심비나 된주 상마 아니 레가치단디 주주니 사숨다 잡시 발라 아니 고라고르데니치 제가 릭직다 레가치 아니 딘 마이베 수국 나로 차와 칸데스 제이나 아니 주주니 섭시 나올 고라고르데시 제가니다 생기 야와체셔요스 제가 단다 아니 가서 두스 제 사제신 바진 델레 인도 참발라 아니 딘데기 아니 콘르도 투두요레스.
It's interesting that 24-hour clock that we work on is probably related to the sun going up and the sun coming down in evolutionary terms. But when you go totally into the dark for a week, if you do the experiment, you still stay on this kind of cycle, and all of your biology relates to that cycle. When you get hungry, when you're not hungry, which drugs work and which drugs don't work. So it's a very interesting kind of story that a lot of biologists study, this circadian rhythm.
제가 겸종게 아니 니주든서 얌셈보 요레스 제가 간주 수부 나올 예베게 아니 다 추주 시기네레 니샤 투직세 추주니 삽시 고라고르단디 아니 당봉일라 아니 고 펜규르 톤 셰비메나 아니 니메케 아니 드로졸 테네 아니 중요레스 임베이나야 아니 다데크 잘케나 dundaji 나우나 루샤비메나다 고 추주 군로단디 아니 무투니 아니 도디기요레스 키라메 지사비메나 메 페마 펜시에 든조 아니 추주니 섭시디나로 카두사마사 그나시 아니 지가 디마임베게 아니 수부크 레가치다 망부치글라 아니 가주 수부 나로를 예베키 아니 레가치다 ki chuzu dik tangzi chi yordas yam sembo yores chesa da dik gyamgyong gyumzen tola tsene ba chu taji mangbo che yores.
So one day, if you're bored and you want to practice your biology of the living staircase, you could think about this, your rhythm clock, and try and understand how it fits into the living staircase at all the different levels.
da khazang anzu ane lobji cheshawa tseso ki kinzik se denzo ane skola samlo tangdo mewa che nyobdo dinde chungsona nyobe ki nimu denzo la ane tanda chuzu ni sabshi roda nisha tuji ki kondu se denzo kola samlo ta nisha tuji ki kondu di ane nganzu ta jengyo ki tanda labshimbi lobze denzo pa gyarvi mbena ane konzu dewa khandes yosare kimzen khachini sare se denzo ane samlo ta yorwas.
Okay, but today we're going to continue talking about our friends, the slime molds.
제가 다 드링은 좀 문투네 아니 슬라임 몰 콜 케제시기스 제가 디 페제 디 나롤 카레
And we're still trying to remember our big question. Remember our big question for the week is this: how does one cell become many different cells?
제가 다 디와 케신부디 카산 란자르다 차분직 차분 링 만다와 망보 칸데스 차레스야 디.
And the way we're going to do that is we're going to start, just to warm up our minds, by talking about the cell, kind of the generic cell and the different parts of the cell like we talked about last year.
그래서 다 당분이랑 아주 다닌 롭티 지신백 내주 가시 텐솔랑기스 안에 차봉세에 가르스기어레 안에 차봉세에 디기 안에 좁제기 차 카르카리어레 세야 된조 다 텐솔랑이 치디스.
And then we're going to talk about the questions you thought about for homework. That is, how do these cells and these cells in the video, how do they change their identity and how would we test that change?
대가난시 다 나원케나서 낭레테 요스틱 아니 디낭롤 요베케 차봉데조 아니 코란케 아니 미지케 아니 콘드스기네 견드스기 디낭롤 코란케 차봉데조 고 겨르고 요레 요마레 고 겨르고 요빈베나 아니 요와디 간조 딱지 칸데스 체투그레 세야데조 삼노단 라이바스.
And the other question was, how do the cells in the slime mold, how do they communicate in order to change?
slime mold se 디낭롤 요베케 아니 차봉데조 펜주케제 칸데스 치기어레스 데와 칸데스 치기어레스 야디 직트와 심바데로와스.
And the answer to these two questions together, the process that these two questions engage, is called differentiation or becoming different.
그래서 다 딘데기 안에 티원이 디기 렌스기어네레 안에 티원이 데다 데와 요베키타 제시 갸단 디 티원이 디 렌게기 아니 네주디 토단 디 and also differentiation sigres and differentiation as the la pressure change lanti gar cheshi change bineti gar so and cheju manda chadre di.
And then finally this afternoon we'll look at our own differentiating cells across the courtyard here. We'll go over and check on our plant cells and we'll talk about the kind of differentiation processes that are going on in our experiment with plants.
그래서 니웅ገብ라 아니 단다 가상도 딱지 체 고주 여러스 딱지 체도 딱샤베케 아니 지싱된 주 나올라 단다 아니 다 differentiation 아니 딘데케 아니 참 레가든 주 카르카르 치기여레 아니 교와 카르카르 두여세 주 콜랑은 주 문준케 시기르다스.
And we'll see and we'll think about, is differentiation in plants really that different than in slime molds? We'll see similarities, we'll see differences, the usual evolutionary thing.
페이지 친구라기를 타봉 그저 아니 뭐 로가로가 차줘야 되죠. 아니 자백 차네 아니 지싱들 주 나올라 아니 제바 로단다 슬라임 몰세 기탄다 가서 무디 나올라 아니 교 로다 되죠. this logares.
So before we start, I just wanted to say one thing about the materials that we gave you, your texts and your the sheets, what do you call them? Slides.
제자자 단다 데르마 만조 응을 키란조 단다 응아조 이제 가시 담소스 롭데비오레 대가나시 아니델 아니 슈구딘데 담도스.
So the way we're philosophically thinking about your learning in this class is that you have a lot of information and materials in front of you. You have the videos that we sent, you have the textbook that I wrote, you have the handouts of the slides. You have a lot of information.
제자 간주기 키란조 담마 테닉 롭디 치다 딩키다 사무당당 토니 지두 잠발라 키란조라마 안조 아니 롭준 체기 이제 망고 쇼치 테시 샤기어레스 아니 얘기 티샤비 롭데데 키란조 도스 대가나시 아니 가상에 망안조 단주 치샤비기 아니 단다 롭제 된주 롱예데니 망보 키란조 따야치 두겐 코리오레스 대가나시 다 단다델 아니 슈간마 밥셰비기 이제 슬라이드 셀렉트 동조 단다 야 퇴양가지 델리릭 망보 파르자지 참 도스.
And so there's two different kind of philosophies, two different basic philosophies to get you to learn that information.
제자다 딘데게 아니 이제든 좀아 담비캅슬 이제든 좀 키란조 롭준 체야 콜 삼노당 인도 삼발라 만조 네다니 돌 삼노당지 이제 담요레스.
So there's kind of a classic approach which is similar to what you use in the monastery, and that is Pantelis and I could get up here and we could just repeat the same information that's in your slides and in your text, right? So we could just show you a bunch of slides that say the same thing as here. So that's one kind of classic way of teaching.
제자다 수준기 아니 롭디 치단 시기네레 제단 페이지 지나 키란소기 아니 공반낭로 롭디 치단 데레 차유 시그레스 소소 롭준 치야 롭데 지어레스나 롭데 담소스 제자 롭데 나 예베키 아니 이게 딘조 이제 딘조 치마치시 아니 간조 델 toya theana shi lobdev ki ani yigja nanglo yobe singdo shisha cheche ma deshe jane lobti cheya chik yores chiko ti chidang rik chik res.
But we have a different, as you've noticed, we have a little bit different approach. Rather than just repeating the information, which in the 21st century, in my mind, doesn't make a lot of sense because you can go on the internet and you can see somebody repeat the information for you, or you can read the information yourself, you can look at the slides yourself, you can think about it yourself. Rather than do that, our approach is to bring our own expertise, our own experiences, and to teach you this information at a higher level through different specific examples.
Cheza da lobdev nang khari yowa shisha cheche ane ma sik chima cheche ti dia di ane nganzu ngun samtang bi bena ane turab nisa ji ki ane lobde chidang sambu chik re sam ki mes. Cheza di mewa songzang ane dinde ma sik khari shisha cheche teche ane labya ki sabla ane tep di nang lo yowe ki ane jodun yingbu khari yinba di ane ngaran su lobdi chini ger su su ki ane nyamnyo da ane nyongwa la jargi cheche ane ma lobdi cheya di ane ko lobdi chidang ta langlentar goya de resam guyes.
So rather than come halfway around the world and just read to you the very things that we bring to you, it doesn't seem to make much sense, right? We could do that over the internet. Rather than that, we want to bring our own personal energy and personal ideas and to get you to start thinking for yourselves about how you can develop your own ideas and your own information. You see, that's a very different way of thinking about teaching and learning.
Zambulingi ene saja chegas dine pine so yung an de le du zambala ngema yikcha nang tisha gyo dma chando yangdo chediwi mena tizu thola re sam gudus dine cheya zamji imbe nang anzo thaja da dine juni chine de ros imbe ne ya di maimba ane del le che ane su ki nyamnyo da su ki ta lobje chidang da dine thone ane ma ta nangdo khariyo di lobgu pe da dembe thon lobgu chebi mena ane ya yor sam gudus.
So it's about thinking and application. So what you have in front of you is 60 or 70 years worth of scientific experience that Pantelis and I have in different areas of research. And we're trying to bring that experience and those ideas to you, right? So that you get excited and you learn to think. And then you can apply those to any information that you encounter.
Kiransun dun mini yoris mini de khares labim bana ta lo dunju mazim ringan la ane tsere laglen tarbe ki nyamnyu ki misha de yordas cheza da denzo ki so ki ane nyamnyu shisha cheche ane ma khachen ta lobri chi yone signere ta zinda tv bina ane kiransu ya jang thubya da ane penzo cha yor sam gudus.
So that's all by way of saying that the slides and the handouts and the texts and the videos, they're all very important sources for you to go and read and watch and think about in terms of what we talk about. So for example, when we gave the homework assignment last night, it would be very useful, I bet some of you did this, you go back and you look in your textbooks, right, and you look at the handouts and you see if you can then take this information and apply it to the questions that we asked, or the questions that you guys came up with that we then asked.
Ta kiransul ije mangbo stam yoros lobde da ane thana shi lonye ta ring mangbo yore cheza denzu sangma ane loya da taya denzu che kapsla ane thanda ngazu zinda thi shabe ki ane lobje denzu da ane durgi ane lobje zinda thi shabe ki ane jushi denzu ki ne ju kol an thi shago songza an denzu samdangi di logo che gu yores chigi gires kasange ane thanda nangle thiwani te sos cheza thiwani de kol samno dan sho sedu sambala thiwani de len gya che tula jik jina kiranzu thanda de lobdev denzo kha che ane ngo yosi res.
So these are the information you have are resources, and then we're talking about ideas that are related to those resources.
cheza da nganzu de ma lobji che di ane jushi chita ba ji kol ane ne ju shi res kiranzu labol yigje denzo khares lavina jushi denzo kol da ane dewa yobeki ane nezu dine ane ma ko igdol ko shabeki ane nedan chadi yores.
Okay, makes sense?
That is was.
Can I add something? However, if you want to ask any question related to these slides as we go, we're welcome to answer any questions. You know, it can be after and so on. So if you get specific questions regarding the material you already have.
그래서 이나야 다 단다 기란족 두스마 이제다 된소 로글로그 체육갑슬라 된소다 대화 예배 대화 된데 예배인배나 대화 된소 gel라 코랄이야 가두께 예배인배나야 아니 가능한 랭게기스.
Okay. So this morning we're going to warm up with an exercise where we want you to draw a picture. This is kind of review. We want you to draw a picture of one cell and all the parts of that cell that you can remember. If you could label those parts of the cell that you can remember.
진짜 고주야 돼 낭래직다 냠도 고주인다스. 낭래드마 진짜 낭로라. 저다 디 카르실라피메나 슈구지 간라 터붕기 리무지 티스. 예말로 장사로와 터붕 디기 낭로라 터붕레 낭로라 왕타 가제요레 차시만다 카르카리요레. 디 키랑기 템바 상마 마디 낭로라 직 티쇼스.
Okay, so we're drawing this cell. Where is the cell? So here's a single cell. So you're drawing one of these cells, and we talked last year. You should have learned the different parts of the cell, right?
다 디낭로라 아니 kartik re khartsung yores labi mena tabung re chadi yorwas jeza da tabung chi nanglo la ane chashi kharkhar yore sobda khane jore longma mal nganzo keja she simba sonza ta tisho shanetso konchosum tabung mangbo the marda shanetso tabung chik therda tabung chik tabung mangbo chobe chokba ki ane thanda ti the marda khasa pungdup the marda chejin khashi pungdup the dibe sorash dukda doste di par muna
It's okay if you don't remember the specific names, but you want to think about the different jobs that this cell has to be able to do.
so da chashi din zo thi chashi din zo ming thi la so ta ming ma dem bim bina an din ang lo la chashi su ki ane la khar khar chi yore din zo samdang shos
Yeah, so look at these cells and think about what they're having to do right now, and you can think about what different parts are necessary to do all these things up here.
de la tabung sto tabung din zo ane den sang ma tabung yim ba song sang ko lega mang bu che di do so da lega din de che la ko chashi khar khar go de tabung nang lo la ane tabung khona ming ma den na chile din zo ma tingi du na chile din zo sul ma thi shos to
Okay, so let's just, as a group, think about what you were just reviewing.
다 gelazzo kiranzo dezen sobe kungoni ya karti shawadin zo ya la indas
Okay, so we're drawing one of these slime mold cells, or it could be one of your cells in your skin. So we're just talking about a kind of generic cell, a generic eukaryotic cell.
slime mol ki tabung chi gine re tegana shi kiranzo ki sobe nanglo yebe ki ane tabung chi gine ane ta chikba chadi gores cha tabung chi tigi indas
So we're going to think about the parts of the cell, and we'll label them here, and then we'll think about what those parts are doing, what their jobs or functions are, right?
당봉이라 tabung din zo la chashi khari me din zo chi thi ane chashi din zo thi sa so na chashi din zo la ane lengen khar kari yor chile khar kari yor ditagindas
Okay, so start me out.
ya la da sta khari yor tabung nanglo la nucleus
Okay, what's in the nucleus?
ane dening nanglo khari yor genes
DNA or the
that's
genes?
rigzela na DNA lao yo DNA de ze khorang ki ming ro
And what are the genes for? What do the genes do?
chesa da rigze din zo khar chi yer lenge khare rigze din zo khar chi yer sen copy what
Okay, and what are these RNA and proteins that the genes encode? Where do these proteins go? What do they do?
chesa da rigze ki ani RNA da RNA ki ani tigar din de tingures tigar din zo khod ures outside the cell.
Okay, some of them can go outside the cell. How do they get out?
Drepung chilo ronye dinde yoro was. Drepung chilo rodang khanis dures. There are holes.
Okay, so there could be kind of holes, but how do they get there?
Chisa ane kola drosik go yana dipadla khanis dures rodang khanis dures suki dures. Huh? RNA. Suki re. It transported by RNA.
Not exactly, but something like that.
Yung mares. This ran dig muscles khanis dures ko chilo thena de khanis dure. Kombazun dure phirne dure modana dure harina dure namruna dure. Kinya sore. Ah? Metaphor. Metabolism. Ah? Jiden tasulana diro khasa organelles.
Okay, so there's different organelles, right? Those are the parts of the cell, like small organs inside the cell.
그래서 drepung nanglo yeki ane chashi simpan ju sangma chashi chik min khala gore organelles lagi wangtalan lagi wa manzo.
And one such organelle is the nucleus, like you said.
그래서 다디 ane dinde nanglo ne mangbo yo nanglo chik di tanda tening la di khona ane wangta chik roas.
And you got the general idea. Some proteins that are eventually made from the DNA, well, the DNA encodes RNA, and the RNA is made here, right?
그래서 당부 DNA 게 당부 제단 티거 디난 수그레스 DNA 균 수그레스 라는 디거 데님베나.
Then the RNA leaves the nucleus and goes to the ribosome.
티거 데 마수가 당부게 RNA 수그로스 RNA 게 티거 수그로스 아니 RNA 게 티거 수샤와디 아니 수잘 두그레 라이보타즈라 디잘 두그로스.
At the ribosome, you make some of these you make the proteins, right?
그래서 아니
So the RNA is translated into proteins.
그래서 다 딩을 라이브 수라들 레주 삼발라 아니 딩을 RNA 유베키 네주든조 라테네 다 디거 차스와스.
So I'll just add to our list here. Ribosomes.
그래서 라이보타즈라는 티긴다스.
These are the protein factories, right? This is where proteins are made.
그래서 라이보타즈 디거 수기로와스.
Now, many of the proteins stay inside the cell and do jobs inside the cell.
그래서 다 디거 렉 망고치 코 타붕랑 낭로 데체 타붕랑 레가 치기로와스.
For example, the proteins are like enzymes that are involved in most of the functions of the cell, transcription, making RNA, making protein. So some of the proteins themselves are used to make more of themselves or to break down other proteins.
그래서 다 디거 낭로 디 낭로 데두 삼발라 아니 코랑코랑 레가 망보 요레스. 만주 카시 구제라 데네 레요. 아니 데네게 아니 든주 상마 디거 그 좁겨로와스. 디가나시 아니 디 낭로라 아니 디거 코랑 요 카시 양마 식치 아니 디거 심바 투베키 아니 주제샤 두로와스. 야 디거 카시 아니 수수 가리요디 아니 디 낭로라 아니 팔로게치 망루 친치 아니 카시 칠로 나야다양 카시를 딜 낭로를 유베클레가 심바드 주체 치기요로와스.
But other proteins, like you said, are secreted or leave the cell, right? And those proteins go through the endoplasmic reticulum, the Golgi apparatus, into vesicles. So endoplasmic reticulum, Golgi apparatus, into vesicles, and then they leave the cell.
타붕 릭 카시 디거 릭 카시 타붕 칠로 등그레스 라로와 다 되니 든조 아니 디낭로 유베키 왕타 딘데 유네 아니 마칠로 등기요스 왕타 든조 아니 카르카리 요레스 라비메나 아니 골지라 소로 든다 드가나시 아니 탄다 디낭로라 아니 페제 시르북란 티유 some vesicles like 디직체 아니 endoplasmic reticulum la 든조 낭로 등기요레스.
And what else?
심바카레 뉴지 타붕라 딜라이다 харсан where you make energy.
What are those called?
Mitochondria.
Mitochondria, right?
Nuji wanta okay mitochondria. I forgot English.
So a very important part of the cell that we'll talk more about later in the week, mitochondria.
진짜 다 누지 원다 세도 타봉 나로네 아니 차시 쳐봐 찍체 제가 다디 타봉 길래가 지당 나로네 개체슈직 임버 손자 다 둔다 나 제도라 가지고 마이토콘드리아 콜 랍기 인다스
Mitochondria have their own DNA, their own enzymes, and they produce energy or ATP.
제가 다 누지 원다 세도 가르라나 코랑 기한 나로라 아니 코랑 기 아니 DNA 요레스 아니 코랑 기 아니 단다 같이 그래서 라빈베나 트리거 된 요레스 아니 코랑 기 칼랭게 칼라는 리버트리언트 체크 요레스
What else?
디 마임 바신 바다 원다 칼라스 나로라 아 골지 수라디 골지 레스 골지 게라 코랑 수라사 심바 아 셔난 타솔라나 셔난 타와라나 디르 칼사데 Endoplasmic Reticulum ladi 심바 cell wall
Cell wall or cell membranes, just some kind of protective
박바다 아니 칼사 페체 치고란티
membrane.
니 was 잡아다 토니
Eukaryotic cells have what you call a cell membrane, but same basic idea, right? You need to protect the cell, and actually all these organelles also have their own membranes.
그래서 다 잡은 거라라 안에 질론 수수 송접 부채들라 박바 도스 잡은 게 기mol라디 그거 당시 아니 리나로게 왕자 춘주 든줄 임베나 그냥 코랑라 아니 덴데 박바 잡다 열다스
Okay, we're missing one set of important things. Let's think about what these cells are doing and how they're doing it. There's a couple of important parts of the cell that we're missing, right?
단도 가시 상마송라스 아니 가르스 콘주 삼로 콘주 내가 지단 삼단다스 내가 지단 딘데 지구유나 안콜로 차시 갈고스나레스 단다 펠로 밍말레주 가시스두스 아 프로틴 spindles I think he's talking about spindles or something.
Okay. What do they do?
레가 같이 그랬어. It is kind of a thread that holds the
Right.
cell together.
Okay, so we call that the cytoskeleton. So cyto means cell and skeleton means structural framework.
그래서 그런 라이드 사이토스켈레톤 라기어서 사이토라는 타붕 라기어스 아니 스켈레톤 라도 삼발랑 아주 루룸 세야데로 아주 파 루고다 되네 그런 타붕 그런 게 쏟다 디 아니 냥냥이 나로라
So there's sets of proteins that give this cell a skeleton, right? Give this cell a structure. And all these different components are probably connected to that cytoskeleton.
그래서 아주 수부 나로 임베나 아주 수부 그 쏟다 칸데 용 마요 세야디 수부 나로 게 루고든 조 치기여 와서 그래서 다 타붕든 조 아니 이 시기네레 쏟다 칸데 용 마요 세야디 디 나로 예베케 아니 단다 쿠바든데 여디겨 레스티자 루베크 라가치니 만족 루고 잡들라
So why do you think that's important?
콘조 온디 가진 계신 분이나스 콘조 든데 게 아니
We're watching these slime mold cells. Why would a cytoskeleton be important? Think of you as one cell. If we use the analogy of one person as one cell, why is the skeleton important?
그래서 다 슬라임 몰든 줄 뜨다스 대가는 키나는 쌈단다스 키나 루고 고야데 가진 계신 분이샤스 it can give us a shape.
Okay, it gives you a shape. Good. What else?
어그 sobda sure 심바 디 소나 칼펜도 여레니 카레 it helps us survive better.
Yeah, it helps you survive. Why?
가다 간데스 펜도 디 소야레 펜도 유는 토톤 라다 it gives us the ability to walk and to move.
Yeah, so when you have a shape, when you have a structure, you can control your movement, right? So I can go like this, and I can move, and you can sit up, and you can get up and leave. So it allows these cells controlled movement.
Ya laya ma deya pa droya ende che dun sama sui kadin re rugo kadin das
So we'll say structure and movement.
structure la dun sambala ko ba ib ane gugye cheza ib la tene gugye ta sobda yuya rugo chigiyores gugye da di rugo chigiyores.
We'll just talk about a couple more things in the cell. One other organelle is called a lysosome. Did you hear about lysosomes?
Laisu suk lai ji kunyu yores kinzo di thanda pa la maswas yores
Did you hear about those?
kunyu yores.
So just think about lysosomes as kind of a recycling center. So proteins go to lysosomes and they're broken down and recycled.
Chesa laisu suk siginare laisu thasu peji laisu suk stiyo kurang ki lenge khala na ane soda ruma sube ane chala ane nyingba denzo ane sawa sugiyo anganso this person nangshi recycle lagiyo anganso denzo khala na dinanglo yewe tigar denzo ane ma shik goya denzo shik ane ya sawa ya dik goya denzo dik dinde che goya yewe me na tigar denzo dosa khwa dures la na laisu suk la dinanglo dures.
Lys means to break, and some is the body.
Chesa da lais la ju zambala konzu khala giyor slabin be na ane tuba ma shikba dinde khutunda res ane som la mangbo yo denzo sangmal ganzu suk la sugyo body sidu zambala sukbu la giyor res chesa da dinde ki ane yib yunye sobda yunye dim ba sonza ane som laisu suk da ane dinde giyor shayor ta.
Okay, last thing, most important thing that you guys are forgetting. Again, if you think of your body as one cell,
Right. Kiran zu jig temba jestas manzu kiran samda das kiran subu ganza tamu jigina.
how do you respond to stimuli? How do you respond to stress? How do you respond to anything?
Chilo ki nezu ji chungbe kapsla nezu dela ane yalen khandes chigiyores da khandes tigiyores kinzu da khandes tayem bes kirangi.
Right, because right now this cell that we've drawn has no way of sensing the environment. How does it respond to
Da
anything?
danda kiran zu ki lache mangazu thisha gyo di ane chi khuryu ki nezu ane sortove ki niba che mindos di gorwas ko kharchigires. Receptor cells.
Yeah, not receptor cells, but receptor proteins or receptor molecules, right?
Dalengi ane Drepung da dalengi ta wangsa da dalengi tra dinde laya mewa che dalempas chesa ta dalempa di ane khashi ane nelempa ta khashi ngema receptor la ngazu nelempa thiyor chikchina dalempa thiyo sir chesa di ane khorang ki pakwe k nanglo la nizu karin di sortomye sornghe lenghe dinde yordas.
So this would be analogous to your eyes and your hearing and your touch and all your senses, right? A cell needs a way to know what's going on in the environment so that these slime mold cells can move around and do all the different things that they do.
Chesa ta ki di khar daures la na kirang ge ane mig da amjo da denzo daur pakwa denzo daur das chesa denzo ki ane kilo ki nezu denzo lengi do chesa ta chilo nezu lengi ane tinjen ku kharchi gyors la bimbe na chashi shemba peju tanje phado gosal phado su yung gosal su yung chi gyores.
And there's all kinds of different sensors in cells. There's receptor molecules that are made out of protein, right? That can bind something on the outside and then change something on the inside.
Chesa da danda dalemba ina re nelemba ina denzo mangche khares la bimbe na dudu dinde chesa da dudu trobek di khashi ane tigari indewa chesa konjo ki ib chiraba chi gyore ib chiraba di yebe gyumzen tene ane ib de dan sambe ke chilo ne jala le yundu sambala koni jargu yore jarwe gyumzen tene ane chi ki nesu di nang ha ku yores.
There's so-called channels,
Gana shi en konjo ki nanglo ne channel sil sambala ya ma dolam de yores.
which are usually made out of proteins that can open and close like a gate or a channel.
Chesa da savek chane ane dolam de zo khari ku thoba des tana ya tigar ku thoba des ko kha chewa da kha sumba inga cheya yores.
There's receptors inside cells because some molecules that are either very small or have certain chemical properties can move right through the membrane, right? and then they'll bind to receptors inside the cell.
야 넬렘바 카시 과데레 Drepung kora nanglo de deores. Kunzu khari lingi res labi bena kunzu leya ki ane da denzo sigune re ze denzo ane Drepung pakwa gyune ane goza dene gyu mowa cheche tega dotu yores. Tega dotu yo sonza nangle ingore nangle sabe jela ane nelemba denzo gis ane lingi res yan simgi res.
So remember, this is just kind of a generic cell, and depending on what kind of cell it is and what kind of organism, it's going to have different amounts of these molecules, different amounts of these things. So for example, a muscle cell is going to have a lot of mitochondria because muscles need a lot of energy.
제가 다 딜라 sample 자백 차네 Drepung yin chinche la korang tumongi ane sugda cheju koba dene do yores. Inaya da Drepung rig manda mangbo yo sonza ane Drepung rig chida ba so gobe ki ane goba khari me dela tagi ji di nanglo yebe ki chashi denzo kashi ma roya da kashi nyung roya ane sobda ane loga cha roya dine yungu yores special shana ane subu ki shane nanglo yebe ki Drepung denzo yimbi na ane Drepung denzo ane tanda nugi wanda mangbo gu yores mitochondria se di nugi wanda mangbo yena ane ko neba dosu mangbo tang gu yorwa chita dishine ane konzo nugi wanda mangbo yo cha yores.
Or cells that are going to be secreting a lot of hormones, they're going to have a lot of Golgi, a lot of ER. So every cell, depending on what its function is going to have different amounts of these organelles.
그래서 다 대가나시 아니 양 지가 Drepung 아니 수국 나로 마 실구 망불려니 그 Drepung 임비나 아니 콘졸라 골지다 대가나시 ER라 든데키 아니 탄다 숙든조 차시 든조 아니 망보여야 차용구 요레스 딱 한다마다 가리네면에 아니 소소수기 아니 제레라 파베키 아니 코란스 나로라 원다 든조키 아니 망뉴다 아니 제바 교두 요레스.
Okay, so any questions about the cell, because all the rest of today and the rest of the week, everything we develop, all our ideas are going to be based on the cell. So we want to make sure that everybody understands the parts of the cell and what they do.
그래서 다 Drepung 토를 되어요나 세우소구여스 아니 단다 드링기 니마자 많이 받아 눈다디 찾아 아니 내주 가르시에 임바던조 상마 아니 Drepung기 아니 내가 찾는다 Drepung 그 도단 실라 샤베 토네 아니 시에대야 장자 차디기요 레스체자 다 당봉에나 Drepung 차시다 Drepung 질레 던조 토를 아니 단다 되어요나 되어고쇼스 되어버리
Drepung 지금 동다다 충충쇼라
Remember, you have to talk loud enough.
충충쇼야.
KKS 잡았어.
Drepung 지금 충충쇼야. 최종의 차가는 충충쇼도. 디스노라는 데 망부 컨디션으로 있어.
Maybe you can repeat it because they can't hear in the back.
Drepung 거라 춘주 쇼시도 Drepung 나로 드네 망부 칸에 슝구여레스. 그래서 cell normally we say that cell is itself is very small. So it is a very small object. How it can have so many different organelles or parts?
The organelles and parts are smaller.
Res Drepung the chinju huge res. Inaya di nanglola dinzo mangbo yego tubia kajin res lana. Di nanglol yeweke wanta da chashi shimbadenzo dile chungo res.
So remember in evolution, remember we talked about how life began, how life evolved from non-life in evolution?
Pengyurge ane ta namsha nanglo la ngazo ane chende mayim be ki ngobo ne chende ba chada khandis yeme ngazo ke shigi yos.
So remember that life probably started with non-living pieces of what would eventually become a cell, right? So when life was evolving, it was starting with molecules and very small things, and over time, over millions and millions of years, took on complexity, took on different parts. So probably what happened, we'll never know for sure, but probably what happened is we started with the small molecules, and the cell was built around those molecules, so to speak.
그래서 다 pengyur namsha nang shidu samba chende mayim ba ne chende imba chadu samba chaya ki keje shigi yore. 그래서 다 chende mayim ba laya dezo ane chende mayim ba ke ane ze dezo ane dudu shunju dinde chadi yores. 그래서 다 dudu ane dinde soche ane dig sokwa dezo 아니 고뇨 루두 삼발라 아니 간조 탄다 아니 지금 고뇨가다 칠레 마하키 아니 젠데 뭐부도 차투기로 와서 그래서 다 타봉세 디코라 아니 디낭로 유베 차든조 아니 델레 충웨키 아니 냐마임베나 아니 젠데 마임베키 아니 제세네데 두두시게느라 아니 덴데 속바 소체 아니 트롭데와 차기 요레스.
But always remember there are exceptions. Some cells are big, like the muscles. Some muscles here that connect from here to here can go up to centimeters long. And frog eggs are visible, and I brought some so you can see them.
이나야 타봉 상만 취주시다요. 쇼 취주케요 마다스. 마조 수보 나는 예외케 샤주 그 타봉도조 가시 되면서 열다스. 안 대하나시 배워다 된조 그 공원도 타봉지로 keramiktan tongtobe gyok tigums chimbo yoris.
Yeah, so again, structure function holds true at the cellular level too. Like squid neurons can be 2 meters long. Others, so they have a certain function. They need to send a quick signal a very long distance, right? Other cells need not be very large. Bacterial cells are very, very tiny. Some bacterial cells are bigger. So always think about structure, function, also true at the cellular level.
The tabunki and Rambal Kejashi Bimbinaya, and a Koranki Koba da Chileni di, and a Deva Yoadi, Ranjani di Yordas, Koranki Koba Kandis Yoma Yusia, Chilel Rally Yores, Scotland, Tanda Sonal, Dediniki, and SMG Gore singing Yeshua, and Zimbina, Konzek wants the tabu mideni Yores. What is that? Is that any tabunkashi and Chinju show is the Peshashana, any bacteria said so do the Zuimbina, testing the Yod, the Zuimbina, and Chinju shows to us. Is that Susu Chilel Power, Langel Power, Kashi Ringbo Yore, Kashi Tundu Yore, Kashi Chimbo Yore, Kashi Chinju Yore. Yeah. Kitches again to Zutola.
Ni toda carayos, Dangazu. 미도지나 도번 타부질 좀 두버라. 근데 타 먼저 돼야기 타분 되나는 땅 먼저 먹어 갈라. 로가 옛날에 샤이나에 가린 참 되게 니가도 참 되는 초대는 소리지 되시지 나서서. 근데 이 놈다 배차셔는 때 미타분인도 지금 게 가만데 게 아래도 지나라. 로고 되게 타분 지어라. 타분이 니가 집에 지라요 우리가 요 말이. 근데 기시 니가도 제가 쇼기는 가진 쇼거를 Gary top chamorus to the and the tabuna
So if we all started with one single cell, and then right now we have different kinds of cells in our body. So can we say that that original single cell has the potential or he's using the word energy, energy to have energy of all the different cells that is present in our body at the moment. So and then the other part of the question is, for example, we say that that original cell has the potential to give rise to let's say bone cells. So, Ari has bone cells and Kama has bone cells. But Ari's bone cells are taller than my bone cells. Why is the difference? Even though the original cell has the same potentials to have different cells and also have the potential to have bone cells, but bone cells become different in Ari.
Yeah, I mean, I think the answer to your question is a very good question, right? The answer to your question is built into the answer to these two questions, which we'll talk about in a minute, right? But at the very heart of your two things. One, yes, we've talked about this before. We talked about this yesterday and in past years. That single seed or that single cell has the potential to become the full organism, obviously, biologically. Philosophically, it's another question, right? Like what exactly is the essence of, is one cell the same as Ari or the same as Karma? Don't know. So we don't know the answer to that so well, but certainly it has the potential to become a full Ari or a full Karma, right? Second part of that is why, even though we are very different, we're also very similar. We're very similar. Almost almost identical, right? So what accounts for the small differences? Color, size, very few things that we'll talk about those in a minute, right? But basically, we're identical. Everyone in this room is basically identical. The differences we see in this room are very small, and we'll talk about where those come from. I think you talked a little bit about that last year also. The answer is genetic difference and experiential environmental difference.
제가 당부 고조스기 처분 딜라 진짜 렌 되었군요. 그래. 야부친이야. 네, 당부 고조스기 처분 딜라 안에 단단한 주 속보 나올 교회에 처분 된주 상마 용식이 안에 콜라 안에 뉴버시기네레 안에 제주 시기는 가르치는데 여러웨스라는 게제티나 렌디 여레스 라오레스 어디서 제가 디마 손자 안에 처분 찍네 강다만다 젠댕 공부 치다바 찍 차토비아기 아니 제주 요 손자 니바 요 손자 아니 먼저 치다바레레 차 연구 여레스 다게 든조 차사베 제라 아니 게 페셔선 게 아리 든조 아니 링부두스 게 까모틴두레샤스 가시니 레스 당봉에는 저분직 나올라 니바 차사 여로스 루고 가시 링부 차고야 가시 튼두 차고야레 라주삼발라 자베 차네 아니 칼라 여레스 라빈베나 만조 상마 아니 저분 릭직번에 조바임바 손자 아니 만조 나올라 자베 차다 만다베 차 찌빈베나 자베 차데 망부시다 여다스 만다베 차데 뉴뉴시다레스 이나야 아니 젠덴직 나올라 매부 찍는 중에 그 처분도 제일 처분 로가 차도야 대가난시 안에 미 리직바 임빈 직반의 중에 그 처분이나야 제일 아니 미 로가 차도야 된조 아니 제와 춘주들이 망보여레스 제와 춘주들도 같이 있는 연구여레스 야 된조 자외기 간도 단다 된다 디글로브쟁 주시레 대가난시 안에 롱웨마 더발라 페제티스티스 라이어레스 김제든도 단다 제일 리다스 제가 딱 구류다 된데 내당 토니 아니 연구여러스
Other questions?
계시신다.
셀리기 때도 자본기 도마 거바테 가르서서는 아마 컴바질에 배단 던져 소리 질러서 이슈
민다요.
다때도 단다때 거바다 실리시우도 다때 코 컴바질렸다네 거 소요 바이나 근데 니 파수 노노를 샤시에 되니 거 가슴 저와나 붙일래 거라 때 간슬래
initially when they look at the cell, so actually cell is the monk's quarter, monk's resident quarter cell.
Right.
And so when they named cell for this particular thing, they may have think about some reasons why they use the word cell to describe this. Because can we take example as a room and then describe its functions related with the cell?
It was a Leeuwenhoek. I think Leeuwenhoek or Robert Hooke, whoever named the cell. It's just all he saw, because the microscopes were it was the first microscope, right? All he saw was something that looked like a cell. He had no idea that all these other things were inside the cell. So he just called it a cell because it reminded him of a monk's cell, but he had no idea about all these other things. That was just the beginning of trying to understand what a cell was, because nobody had ever seen one before.
Again, tinjezo tabungse dela injinala cell di tasha sigero tasha kangben tabungla tasha kachin tares ladu sambala chanton cheshi ola tanye thangbu di koran te edu sambala di nanglo chashi de ton tube ki chanton cheshi ji yorwesla na yomares chilo pagwe sobda de jikon tungu yores koran thangbu del teve kapsla ane tupsi de mangbo yowa sonza tupsi mangbo de tongdu samba koran khat thene oh tasha ndala samresko sobda di mo sonza dinzu min khalare tasha da de chomba dinzu desa yang khangbo tupsi chunju de do khanda mata khangmi chunju chik jil chik tune gya sha gyo de dawo yowa sonza khalare sel di main be shimba jumze yomaris min ta sha gyo
무슨 서울 감독부터 차봉되고 아니 그 리가 차선 찍어요.
아?
서울 감독.
예. Drepung nal yewe ki any cell drepung sabun nal yewe drepung ki lega de chigi welas. So the cells present in a dry seed does all the has all the function all the parts, and do they do the same function at that stage?
Okay, good question. So this gentleman's question is, if you're in a dry seed, like the seed before we added water to it yesterday, just a dry seed, the question is, do you think it has all of these components in it? And if so, what are they doing, right?
Yeah. So that gila kurang gila di suyang kambu la di zambal khasang anju suyang kambu tabimbas pe chumalu ngi suyang de ruas. Chisa dig nanglo la ani thabung dinzul chashi dinzo yorebes. Yona kunzu ani lega chigi yorebes. Khandis chigi yores ya dires.
What do you think?
Kirans kharsam gidus. They have the parts, but they are not doing any function.
Okay, so it sounds like you're saying they're there, but they're somehow taking a break, right?
Chesa da dila chashi dinzo yores ina lega chigi yomari la zambala kunzu parsing de yores yorwas. Yes.
Okay, so that's your hypothesis. How do you test it?
Chesa da di chöba imba chashas chöba ina da de chöda khandes chior daji khandes chiores. We have to sow the seed. We put water.
Okay, so if we add water, like we did yesterday, and the seed grows, how do we know that the mitochondria were already there and then became active, or weren't there until we added the water and then appeared? So we haven't, that test doesn't answer the question. Remember, our hypothesis is that the mitochondria are already there, but inactive. We add water and they become active. So your experiment doesn't quite work because it doesn't tell us if the mitochondria were there before or after.
Chesa suyan tab ne tagi isla da chulu ne tagi isla sambala thamung ne suyan de ne ya chik sabu chele gi nyugu chele gi na dige kirang thamung ge soba dela ani leng ge mi dos. Soba nanglol kharis la bim bena chashi din su sabu nanglol yores ani jela dirizamba person chin de res larwas. Tada da nyugu di che kapsula ani nyugu che di sambala din nanglol da nyuji wanta lega chigi yorwa. Ina nyuji wanta de kirang chulu ne chunyam do le de sabu khona na thamung ne ye di ani nyugu che gyumze de ne kirang khanis che turis.
So what's an experiment? Can you come up with an experiment that would tell us, are the mitochondria there already or only after we add the water?
Chesa cheju zambala ko chewe lega chigi yo sonza nyuji wanta yore sigirwa yogurwa. Ina ya nyuji wanta yo da chunyam do le imba da ya mena thamung ne sabu nanglo yo ma yo se di kirang yawa khandes chugres yawa chewe takje khandes ya imbes. We can check the water whether it is clean or not. I think they mean that whether there is mitochondria or not
In the water.
in the water.
But
What? Khandes how? Je jigra. Chena mitochondria yoro ani. That sabun nyugu chigi yuna mitochondria yore si word dina. Sabun nyugu chigi yuna mitochondria yorwa sabun lega chigi doa. Chesa di mitochondria de sabun khona thangbu nge negabla kambu yimbe negabsla yogyo rewe chunyam do legyo yime di yawa khanes chure sabun sabuni tawa da sum tawa chikbarwa. We have to check the water. We can boil the water.
okay,
And then boil and then let it cool and then put it into the seed on the seed.
Okay, let me ask the question a little bit differently.
Kilaso tiwa de go chugi samaris kuidang mandas kuindasta.
So the way I interpreted your question was, which situation is happening? Either there's a seed with no organelles, we'll just focus on mitochondria as an example, and then we add the water, and now mitochondria somehow appear, and you get a plant. This is my bad picture of a plant. Or mitochondria are already there in the seed, and we add water, and it somehow activates the mitochondria, so now we can grow. And my question to you was, can you come up with an experiment that'll differentiate these two?
Ya pochi nya, ya pochi mani tiwa go mazu ko ni lenga ni mo maja. Ne dan ni te yo res lar da. Ne dan khares la bi na. Drepung kambu di imbe kapsula ko ko nanglo la. Drepung de nanglo la di ke wangta de je me wa che. Juma ane chulu ya da sabu da ma tabya da dinde che kapsula ane ya che yum du zambala ane di jela ane shine che ne kapsula kambu imbe kapsula mewa ane jela ane taba da chulu wa dinde che du zambala ane wangta de le de gyo dinde che rewe ya me na ane drepung de kambu sun de kambu imbe kapsu imbe da ya ane korang nanglo ge wangta sangma yo ta wangta nanglo pe chira ba gela ma ju konde sha so nuji wangta sha so nuji wangta thangbu ngone yo wa che 이내야 디젤 출루에다는 살 답이야다 dindeso chidu sambala ko nyugu dia chele yowa 제자다 디니니 내단 가르 라두 sambala 가리 이메디 안 간주 그 초다 간데지 치나 아니 내단 디니니 가리 임바 간데스 싱게레스 초다 가리 그 토네 강디니 오디니 싱게레스 등이 현안술 파티오고 있스 제자다 가리 인서 갔다 다지 간데스 치나 디임바 디임바다 디 마임바 야메나 디임바 디 마임바 간데스 하고 그랬어 다지 쇼쇼 삼다니 아?
성감부지다 성분바지 니지
사불롬 롬발란
난시르게 시들이 니지바 감분나니 한무시게 따니 나고지 트레이너리어라 몸반나지다
we can look under the microscopes. We can have two different cells. One is completely dry, like a stage. Other one is a normal cell, and then check whether there is mitochondria or not.
Great, perfect. Then we know the answer, right? So if we have a way of, say, staining mitochondria, and we stain the cell, the dry seed, and there's no mitochondria, right? Then we know that there's no mitochondria. If we stain the dry seed and there is mitochondria, we see mitochondria, well, then we know before we add the water that there's mitochondria. Then we can do the same thing after we add the water and check. So perfect.
So that 먼저 같이 둘러나 mitochondria 뉴지 원터라 저 카레스라나 게라 코랑 라우나 시타도 제시나로 톤 투에 여베기 아니 원터든데 다스 제시 그래서 딜러 선발다 당부 사부의 깜부 인기베기 네갑슬라 찍되스 주마 안에 럼바 차제 제사웨기 네갑슬라 찍되스 닌가 나로라 누기 원터 누나 안 누기 원터 가둘리 요네레 당부는 요 아니 출루는 레와 마임바 제사다야 따두 점발 당부 깜부디 나로 메와체 아니 제라 제사웨기 아니 타붕든 쭉 나로 누기 원터 요데와 이나 아니 누기 원터 가둘레레 chuluwada suen tabe jela chuda ta sada khaluna dinzun legyo imba chadu ruwas.
Very nice.
Oh, keran reasons.
Which do you think is right? What's your intuition?
Jaza ta takje chena dine ha gudo ta keran te bin bena manzu gang ni sare od ni sare kis di this one this lower one.
I think this one.
당문이는 딜라인발. that dire khar ta thamadinda dire gandere odere. Maybe lower one.
Do you think both? Which one? It's only one or the other.
That ya gandere lab odere layo mardalas. Ni yeme Chigmishi Yomadas. Yes.
Let's take a poll.
Yeah. Gela number Ninjitsu. Nuji Wanda da Dunzo Tama Tambungu and the Tabun Kambuti capsule Yoris. Chule Lady Gomares, Lanya Tama Lawacham.
They're voting on this one?
Tamasha. Nuji Wanda Tambung in Yomares, Sabun Kambu Bicapsula. Cochuda, Salu, Yell, and Chile Yore, Lanya Dilawacham.
So why do you think it's this one?
Ta manche dire sigdo di yi go gyumzen khares dire lani jik shoda
tambo dina yo kana sila cheta mancho ne ko de
koro dio koro lengi so it is there right from the beginning, but since we said that they are taking a break, so that means they are not doing anything. But when they are combined with water, then they started to function.
Yeah, I would guess that's true.
Can I ask a question? So, what do you think? So, your mitochondria, right? So, you're like kind of seed of embryo. Where your mitochondria come from? Your both parents?
그래서 다 근데 임베나 타봉 당보니 유교 임베나 계란 미로 임베나 아주 그 타봉 통마디 파만인가 뉴용교로 파만인가 뉴용로 그래서 다 디기 뉴게 원다 된조 파만인가 뉴용하르베 파마치다 바칭 뉴용하르베 아
Let's take a poll again.
야 요슈로인다스 파만이 뉴게 원다 된조 파만인가 뉴용레라니 상만 나고장
Second is, if they come only from your mother.
Mares, Pawan, Pamaningan, Yungumares, Amajibun Yungri, Lani Labojan. You get one the Lavorda, Shambachilla, you get one the Layurda, Amajibun Yungri Lani. Oh, no, nobody.
No. What about the father?
Palajibun Yungri Lani.
They all come from your mother.
Tinjenso. Pamaningan Yungu Mardas. Amajibun Yungu Yores, you wanted to do.
And so this is important for genetic studies, tracing, so the lineages. And they don't change much, so they can classify based on that, where people come from, the different.
Just that the reason they do you na and tabung nanglol give nuji wanted him do amachipun yung do amanchipun yung digio sonza and kunzuki and tanda midinzuk nanglol nuji wanted ki and dodanda chagman the zul teche or midinzu riktikba kawa redo kani yung sha dinzu haku yores digio maro.
Okay, very good question. That was a very good question. Because we got into a whole discussion about how do we do experiments, how do we think about solving the problems. That's a very good question.
Teoya udus teoya di kadin tienchi tanganzo takje cheche ane gangne dan khari me se de so da khandes chiguyo me se dinzu mangbuji kola ane nganzo ma droya chungsuas.
Other questions about the cell. Remember, we want to understand what this cell is and all the parts and jobs.
제가 안에 타bun골 탄도 티와 US 타bun고랑 나놀 유베 차시 된 적이 칠레
Yeah, Losel.
타bun으로게 타bun 찍나라 안디 다 두두리미라 몽부슈다게 두두리미라 몽부디 당본이 촉과집으로 양 가니게 가 간데차레스라 두두리미라 몽부슈다게 당본이 촉과찍니 예와레 아니야 다디 시다니 차바레스 간데레스
니만 펜규로 당본에 단답을 라고래 미직 나올라고
타모치그랑 기다나 두두 미라 몽구다게 마이토콘드리아 레조섬다 몽구다게 진짜 당부 타본치그랑 당부 촉과직니 엔카두스 게어레 그 당부 간데스니 차르스게
타본직 닐 차도들 레예 This good. Marta Tabung Kejashina and Tabung Toma Mebu Tabung Tomane Tandapadu Rodan Tamuil Kani Chungri Lachik Share Mijik Subuchik now Yevik Tabun Chidapa Chila Kokani Ledger on the collection.
Tabun Samara get the Chesama in into that you Mito Mambu de Ani Tabun Chik Tambo Chik Kagachare and get the Kandacharis.
So I think what he is saying that since we have different kinds of cells in our body and each of these cells has different organelles. So the organelles in these different cells they came from one single cell and then divide it and get into these different cells, or each cell has a different source of these organelles.
So good question, but so in general, each cell is going to produce the organelles that it needs. But every time a cell divides, depending on when in development it is, it has to provide all the potential for the organelles for the next daughter cells.
Tabung Shimbaji Goshetene Chekachane Tabuni Chawiro. That Chasimbe and Tabuni the daughter cell Legio, Tabuni Dings Nangola, Cook Nangola, Wanta Guyuna, Wanta to do some Tamu in Kwani Yungure. Tabu Amadin Chungura. Jizata Amajina Chungsav Jela and Dinalo Wanta Chidaba Rere, Tabun Kona Nalo Sawasuya, the Aning Bashiki Sawasu Chiguro. Just a dinner Dinal you want a decon Yunlana, the Koran Nalo Suga Chaswa, upon Ding Amadinima Yungur Rebidoa. Inaya ko dinalo so to be a new one lady. Tamungane Yuvik and Tamu Amadinima lady was. What is it? Dini Chairo. Dini Chan Tabu Sawa Chairo, Tabu Shimba Dinalo Yuvik, and a column Mantaya, and didn't want to so to be a new body, and a Tabu Amadin Yungu.
But it's a complicated question. Remember that each cell has virtually the same DNA, so that means each cell has the potential to make as many of whatever kind of organelle it needs. So that's one thing.
Just that Tabung did someone and so Goyaki and wanted someone so to be a key, Nuba Handisli or Residu. Tabung did someone and Rexit Chiba yours. Rexit the Amadin Yungurua. Just a Rexit Yoa Sonza, Rexit de la Th and so Goyaki and wanted Karim, but the Sugi Tabung Susus now Sugi Chigres.
But each organelle is a little bit different. So, for example, mitochondria, like Pantelis was hinting at, mitochondria are kind of like their own organism inside our own organism. So when a cell divides, it takes all the mitochondria in that one cell and distributes them into the two daughter cells. And then each mitochondria can divide by itself to make more in each of those cells. So mitochondria are a little bit weird.
Mitochondria wanted this Manda Yores, Nuji wanted Koran Subuk Nanglola, Kokona and Simjan Chadabachi Imbado, Koran Nanglo Rexios. Just a deem was on that's Tabu Amadina. Amadu Goshet to Zambala, and you cook Nanglo, you wanted Goshetata Tedures, Chakapado, Chakaso Yungres. Just a Tabu Sawa Dig Nanglola, Nugi wanted Tanda Amal you, Chakachaswa. In a cook Harchigre, Nugi wanted to do some Susu and the Palo Gachi and Tanga Kaze Goa, Digam Stick and Sawa Susha Grecia. What do you say? Palo Gasa Hojil and Amakashi Tamun Euro.
Every organelle has its own weirdness. For example, some cells have more than one nucleus even. Some muscle cells have more than one nucleus. Other cells have no nucleus at all. So to answer your question, it's very complicated. It depends on the kind of cell, the time during development. There's no easy answer.
Just that's the Tabunji now you wanted to Tama and the cover chungus like somebody and did that slash Capuyores, Susu and Dinalo gave wanted Rick Manda, Susula, and the Jetanda, Yonta Manda Chadi Yores, Tabuina and Tabunkashila did do chick me, but they do unique Tabu Yores. Tabunkashila did do what I mail the day Yores. Just that is all and Tambungela and the changes there, Tabu Chigni and Nasolia and Gurim Nanola and Gowadrotanda and Chikmanguchi Rally Yores.
tani tanda di Drepung nang la ko chashi ta sama changwa ina di shenang tawa si jiyo ra Drepung nang la shenang tawa la de ani ko saung kambu nang gi Drepung tin nang la ani shenang tawa yo ni shenang tawa yo ina ko la shiri yo bora saung kambu nang gi Drepung tin nang la shiri yo ra yo.
So if the dry seed cells in the dry seed, they have all the organelles, then they also should have this endoplasmic reticulum. So when we talk about endoplasmic, it should have the plasma or the liquid part of the cell. Does the dry seed has liquid in its body?
Probably some very small amount of water. Yeah, it would have to.
You gures manual madebege any chugi do you gures.
Did you have a question over there? Did you have a question?
Yeah.
Tanda di tanda ngazu di tanda Drepung shi gora. Ngazu naza damla yungora. Naza. Naza damla ko Drepung da ko damla di ne kenshi ko rela. This men shi gora da gera di men sin da. Tanda ngazu men dus ki ngongo jejera. 나서 담을 그런 타분 담을 뒤치고라. 매드스 간제스 고르스. 타분 뒤치샤지 매드스 간제스 고르스. 그런 맨 나로야 타분 요가 야나 아니 매드스 양 제로 된 지지는 타분 세로 된 거가 매야. 맨 그런 나로 캡처드스 간제스 타분 요가 요 말레스.
When we talk about different disease, it has to be related with functioning of the cells. And then we also have different kinds of medicines to cure these diseases or to prevent these diseases. So how these drugs can cure or affect cells? Is it because that the drugs themselves are made up of cells and so these cells interact with the cells that you wanted to make changes? Or is it the drugs are some other chemicals and somehow it is affecting the targeting cells? If they are drugs, then how actually it affects the cell, which is a little bit different from the drugs themselves?
So that's a brilliant question at the heart of all of modern medicine.
Da David Tiwa, da so sigunere Tiwa Kichi did the and Tiwa Nidun Shagures.
Two things. One is all drugs have different mechanisms. So the answer is they can work, they could be all of the above. They could be working on particular cells or particular chemicals. It could be chemicals, they could be, drugs work in all different possible ways. So the answer is yes to all your ideas, plus more. And the other is, later in the week, we're going to ask you to investigate carefully one particular disease and the drugs that are used to study that disease so you can understand one particular disease in detail.
The medu sambala kerang shegyo denzu sangmare digang tandon yores me kashi ane ring naso yodewa kashi ane zel choba kashi ane tanda jendeng ki ane dun kharsada tabung da damanda tubdewa kesa kunju ki ane kerang ki chotab chese ki ane tabung del yimbe ya ane chawa chedang da chen chedang di ane manda mangu shu jores chesa di me naza su la men su ki lega chedang da men Suzuki any tanga ngo sigune re tubje logo yodigores kesa tham duna thama de la keranzo nganzu ki naza chira ba chik kolla ane mixi ki ane lega chitang khande insare ane ko ki ane chothab khande sinsare dik mengi ane naza dik tingla lega chitang khande sinsare dik kolla keranzo cheba cheju is.
Okay, one more question before we wrap things up for the early morning.
Tiwa thama dindas tiwa thama suimbes.
Let's get this gentleman because he hasn't had a chance to ask a question.
Koga maranjo dindas.
Ngaso ndwa mita ina tambu socha Drepung chigden ya ta de Drepung mang de tene sochan thoram sochan ndwa mi chare la uyaro. tendina tanda yushin be chapon jang so maung bana and chapon mountain that tenia do so chat and do a million jura cook up in amity lamdo the other is raw. You were thinking also tanda do me the Tata Tramuchik the Lea Hotel stand at the Mambu Niba in Chayora. The Tanda Matun Zamali the Galaman Tenichik, the Andotinian Mamba meet Tandali the Mambu Niba Gentinichi Chagres.
If when we look at the evolution of living things, we came from a single cell and then we become multicellular, and then ultimately we have this very complicated human life. Is there any possibility of the single cell organisms that is present on Earth right now will eventually evolve into multicellular organisms like humans in the future?
What do you?
That is the first part of the question, and the second part of the question is right now, as a human being, we have a very complicated structure and function, and we came from a single cell. So, if in the future, if we keep on evolving, does it mean that we will become more and more complex, and we will have more and more kind of complicated functions in the future?
So the first question was, can single cells that are currently on Earth evolve into more complex things? What do you think?
Yes.
Sure.
Standard Zambulingal you Tabung Chang then the Mahumba Dylan Gonyo, and Tabung did the chassis chires.
And the second question was, can we, as evolving organisms, humans, become more complex or keep evolving in other ways? What do you think?
Tioniwadi, Tanda Midenzo, Mahumba Lamu to do Spingo do Devi Bena, and the Karchasaris said there was Kerang Devi Bena Karchasaris. Not so sure.
Yeah, I mean, the answer to both questions is yes.
And the
reason is the same for both answers. Right? Because, so can you think about that? Why do you think that organisms, all organisms on Earth right now, why do you think that they're still evolving, whether they become more complex or whatever they're going to happen? Why do you think that they're still evolving?
Maybe because of the environment?
Yeah. I mean, by definition, like we learned about and when we talked about evolution over there in the monastery 2 years ago, by definition, Darwin realized that all organisms are always evolving based on the environment. They could be becoming more complex or more simple. Whatever gives them an advantage in any particular environment over time is going to happen. It's never going to stop.
Mutun Rodigres, the Rodio, the any Loning Sokan Gal into Zamba Shambas, Taiwan K and Azu Mutun Pengur Rodi, Hajilana, Manzu Mitun Kuriuna Nedi Yoros. Chesata Kuriuna Nature, Pengur the Rodu Zambalo, going or Durbe, Yamena Chigam, Tanda, Chedanki, and Malela Drake, the Lake Chire, Malaysia di, Sutachurus Lan, Kuriu dig Nanglola, Pendo you make Tachu Yoris. Gonyor Rodu Zamba Pendo Yuna, Gonyor Chol. And you are do your young depend on me bena dojo limbena dojo chola and you do this chesa candes drum and rodin kuriu tachu rimbena dre imbadi young kuriu tachu this thank you study.
So these are all great questions. It shows that you're thinking.
Oh Tio Yabu dos dilla some can labor the Iran could use some of this something use this. Make the Nikudine.
So what we have to do after break is take all of this stuff we just talked about, because what we talked about is all the capacities that a cell has. And then we have to overlay it on these slime mold cells as they change their identity and figure out what's changing over here in these parts to make these guys change what they're doing.
그래서 다 진짜서 디 젤을 가는 속된 젤을 가지고 갈라 이그레스 다빈베나 차분 찍고 가지고 시우시에 사인 바스 망고시 도스 차시 망고 젤레 망고 도 대수 상마 안에 델 예베게 슬라임 몰 레드 키나 카조 약만 가려는 직티시 그래서 오 어디 다 진짜 슬라임 몰 레드레다 어디 그래서 데 차분 레레레스 그래서 다 딘데기 아니 차분 레레라 체주 등음스 유되니 키 아니 소자 된조 냠도 루루 체체 아니 단다 공우직 차루도 차상 간데 체규나 갈 수 있는 체규나 어든조 토할 아니 가도 제르게 제시기래 그나시 단다 삼상 쇼스
Okay, thank you.
소제 퍼센트